Okay, so for today's lesson, we're just going to be talking a little bit about the vision system. Okay, so we're just going to start with a little bit of anatomy of the eye. So first off, we have our lens here, and that is just going to direct the light back to the retina. And then we have our iris, which is the colored portion of your eye, and that's going to determine how much light can enter that lens. And then our cornea is this outer protective layering of the eye. I think cornea like cone, and it's kind of a conular shape here. And then our ciliary muscles here are muscles attached to the lens, and they're going to change the shape of the lens to determine how much light can be absorbed. And then back here in the sclera, it's just another fibrous, dense protective layer of the eyeball. And we don't have it pictured here, but the retina is located back in the back of the eye. And then these extraocular muscles here are going to be the muscles that actually control and move your eyeball. So if you're looking up or down, your extraocular muscles are going to be doing that. And then here you have the optic nerve, which is just going to send all of those vision information from the retina to the brain. So now we're just going to get into more of the specific anatomy for the retina. So again, the retina is located in the back of your eye here. And like we said before, the function of the retina is to take all of that light information from the lens and turn that into an image, and then send it to the optic nerve so it can go to the brain. So just a little bit of the different anatomy of the retina. So here you have ganglion cells, then your amacrine cells here, and then horizontal cells and bipolar cells. And then you have two types of photoreceptor cells. You have your rod cells and then your cone cells. And so rods deal with light differences and cones deal with color differences. So here we can see these two arrows, one for light and one for vision info integration. So basically, obviously your light is going to pass this way, going from the ganglion to bipolar to the photoreceptor cells, but that information is going to get integrated in the opposite direction from your photoreceptor cells to your horizontal and bipolar and ganglion, all of those cells. So how it works for vision integration, again, you have these two types of photoreceptor cells, your rods, which deal with light, and then your cone cells, which deal with color. And again, that light is going to be absorbed, and then it's going to send that information to your horizontal cells here. And those horizontal cells are just going to integrate that information and send it to your bipolar cells. And then again, bipolar cells integrate that information to the amacrine cells. And then the amacrine cells will integrate that information to the ganglion cells until eventually that information will get integrated to the optic nerve and will go to the brain so that image can be done developing. So here on this slide, we just have a nice table just to help you understand the basic concepts of the effects of light. So we're going to be comparing light conditions versus dark conditions. And my recommendation in your bio and your science classes, you're going to have a lot of information that you're going to have to remember. And so I say work smarter, not harder. So I recommend you just memorize either the light conditions or the dark conditions, and then just know the other one is just the exact opposite. So starting with our light, whenever there's light, you're gonna have a hyperpolarization in that membrane potential. You're also, instead of that cyclic GMP, you're just gonna have GMP. So that means your sodium and your potassium channels are gonna be closed. And then it goes from cis and coiled to an all-trans retinol with a straight cytoplasmic tail. And we'll kind of talk about more of this in the future slides, but I just want to get the basic concepts down. And then for the dark, again, opposite, think dark, depolarize. And then you're going to have more cyclic GMP. And then your cyclic GMP is actually responsible for keeping the ion channels open, so that explains if you have more cyclic GMP, that's why our sodium and our potassium channels are open now. And then you have that 11 cis retinol with coiled cytoplasmic tail. In this slide, we're just going to go over the anatomy of those photoreceptor cells of the rods and cones. And then again, like we said earlier, rods light, cones color. And the anatomy is pretty similar. The rods just have a more rod shape and the cones are just more conular. But a lot of the anatomy is the same. So you have your outer and your inner segment, which is separated by your connecting cilium. And then you just have your synaptic terminals on the ends. So now, diving into the microanatomy of those rod cells. In these rod cells, you're going to have a modified plasma membrane disc. And in that, a protein called rhodopsin is found. And rhodopsin is just a 7 transmembrane domain protein. And at a specific amino acid, lysine 296, you're going to have a retinol. And so whenever that retinol absorbs light, there's going to be a conformational change, which then changes the rhodopsin protein. And so that's how we go from the cis coiled tail to the trans straight tail with the presence of light. Okay, so after that retinol and that rhodopsin protein absorbs the light, it's going to go through a G protein pathway. And so in general, with G protein pathways, you're going to have a G protein, which consists of an alpha and a beta gamma subunit, and that's going to become activated by GTP, and then whenever that happens, that alpha subunit is just going to dissociate from the other subunits, and then you're going to have a target protein that is then activated by the G protein to carry out whatever function you're needing to carry out. So in this case, again, light is going to activate that retinol inside of the rhodopsin protein, and then that's going to activate your G protein with the GTP, and in this case that G protein is called transducin. So then the alpha is going to dissociate from the beta gamma subunits, and then it's going to go on to activate your target protein, which in this case is phosphodiesterase, or PDE, and that PDE is going to turn that cyclic GMP, which allowed the ion channels to be open, and turn it into GMP, so that those sodium and potassium channels are going to close. And so that's how that process happens. So now we're just going to go into a little bit more detail about the different types of bipolar cells. So there are two types, on and off bipolar cells. And the on bipolar cells are going to depolarize in response to light, and then the off bipolar cells are just the opposite, and they're going to hyperpolarize in response to light. And then another little fact, just light causes the photoreceptor cells to release less glutamate to these bipolar cells. And then we have these pictures here, on center and off center. So these are just referring to the receptive fields. So here on center, you're going to have your on bipolar cells in the center of that receptive field, and then your off are going to be surrounding that in the peripheral region. So if you were to shine some light on that center region, your on bipolar cells are going to be there, so you're going to have a depolarization in your membrane potential. And then on the other hand, if you're going to shine light in that peripheral region, then you're going to get a hyperpolarization just because you have those off bipolar cells present there. And then just the opposite for the off-center receptive field, your off bipolar cells are going to be in the center, as the name suggests. So if you shine the light in the center, you're going to get that hyperpolarization. And then if you shine a light in the peripheral region where those on bipolar cells are, then you're going to get a depolarization. And I think that'll be all for this vision lecture. You can go to our Instagram where we'll have a quiz to test your knowledge and your understanding on this topic. But hope you guys have a nice day.